Hello, everyone, and welcome to today's Keynote webinar titled Companion Diagnostics, Technical, Clinical, and Commercial Challenges. This webinar is a part of the 11th annual event in the Genetics Week virtual event series. I am Kaylee Bach of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots. Before we begin, I'd like to remind everyone that this event is interactive. We encourage you to first participate by communicating with other attendees using our live chat feature during the presentation. You can find the live chat located at the left of your screen. You can also participate by submitting as many questions as you would like during the presentation. To do so, simply type them into the Ask a Question box and click Submit. We'll answer as many questions as we have time for at the end of the presentation. If you have trouble seeing or hearing the presentation, click on the Help Desk button located at the top of your screen within the navigation bar, or from the lobby within the event. Finally, as a reminder, this presentation is educational and offers free continuing education credits. Click on the Continuing Education Credits link located in the Abstract tab from the menu at the left of your screen and follow the process to obtain your credits. So let's get started. I'd like to now present today's speaker, Stephen Little, founder of Yourgene Health. For a complete biography on our speaker, please click on the presenter window that is located directly below the media player. Stephen, you may now begin your presentation.
Good afternoon, ladies and gentlemen, or good morning, depending on where you are located in the world. I'm in Yorkshire in the UK. It's the evening here, very pleasant evening. I hope you find this talk interesting. My name's Stephen Little. I'm a scientist, and originally a microbiologist, though I became interested in genetics and then human genetics, and I've been involved with DNA and DNA diagnostics primarily for many a long year, probably too many long years. Over the years, I've done quite a bit in terms of companion diagnostics and personalised medicine. So what I'm going to try to do in this next half an hour or so is just to talk a little bit about some of the things that I've observed in terms of companion diagnostics, and then what are the challenges in terms of translating tests and drugs into clinical practice, and how do we overcome these? So if we go on to the first slide, personalised medicine. Now, there is a plethora of definitions of personalised medicine. This is the one that I write and I use. It's quite a narrow definition: the use of a companion diagnostic test to aid in the selection of patients most likely to respond well to a therapy or other medical intervention. And the key elements there is that there's a treatment or a therapy, but there's also a test that's closely linked to it that's designed to predict response. I've got a couple of other definitions. This one's from the European Commission: a medical model using characterization of individuals' phenotypes and genotypes for tailoring the right therapeutic strategy to the right person at the right time. Now, I think the European Commission made a good point there, that sometimes when we think of personalised medicine, it tends to get tied up maybe too much with DNA or nucleic acids, and it's certainly not that at all. There's nothing special about a DNA test. It just so happens that a lot of companion diagnostics are nucleic acid tests, but there's no reason why they should be. The FDA definition at the bottom there is a little bit broader: an innovative approach to tailoring disease prevention and treatment that takes into account differences in people's genes, environments, and lifestyles. So they've kind of broadened the definition a bit to include I guess, screening and early detection. And I'm going to touch on that a little bit towards the end, but mainly I'm going to be talking about companion diagnostics. So for several years in the 90s, I worked for AstraZeneca, but I didn't work for the AstraZeneca pharmaceutical division. I was in the AstraZeneca Diagnostics division. And it was quite a small group, fairly innovative, I think. And we were interested in genetic testing. And, and funnily enough, in the early 90s, we couldn't do very much genetic testing because there weren't very many genes to test for until the cystic fibrosis gene, the CFTR gene, was discovered. And we developed a product to test for that, which is actually still available, or a version of it is still available today. So that was how we got started. There was a lot of talk back into the 90s about, how wouldn't it be a good idea to take a diagnostic and use the diagnostic to get better outcomes. And I thought that just sounded like the most obvious thing I could think of. I thought, why on earth wouldn't you do that? In fact, I was so taken with the idea, I decided to leave AstraZeneca and set up this company, DXS. And we said we were a personalised medicine company that meets the needs of the pharmaceutical industry for biomarkers and companion diagnostics to support the development and sales of cancer therapies. So I think we were in the right place. Maybe it's a little bit too early because, as we started trying to interest drug companies in this, our service, we came across, quite a few barriers. Maybe the biggest one was, we don't need a diagnostic. We're quite capable of getting our drugs to all of the patients, and that's the model that we like. And you need to remember, in those days, that was very much the model of the drug companies. One size fits all. So there was some resistance there, and the other resistance, which was misguided in retrospect, but it was certainly true, is that if you use the diagnostic, and then you said some of the patients should have the drugs and some of them shouldn't, it would mean that there would be less patients getting the therapies, and that wouldn't be economically viable. I mean, if you think of the counter argument to that, what you would be saying is, well, what we'd really like to do is sell as many drugs, and if that means that some people get them and they don't work, well, so be it, at least we get paid. I'm not sure that was exactly what the thinking was, but that's kind of the implication. The other things was that it was too difficult to do because there was the pharmaceutical industry that have to rely on the diagnostics industry to do this. They didn't really know how to do it. And also there wasn't very much precedent on it, or there was no precedent. So the drug industry is quite innovative, well, very innovative in some respects, but in terms of business models, it isn't always the first to adopt something new. Anyway, we set up this company and we were getting on with things and doing bits of business here and there. But I think the thing that really made the difference to the business and kind of set in train a way of working between drugs and diagnostics, which has continued to this day, was what I'm going to call the KRAS story. And I know it's a few years back now, it's 2007, 2008, but I'm going to go through this because it's sort of quite illustrative of some of the lessons and the challenges in terms of getting a companion diagnostic to market. And I think we learned a lot from this. And I think this model that was set up in this project has been repeated over and over and over again between drugs and diagnostic companies. So in 2000 and back in 2006 actually, the FDA approved a drug from the drug company Amgen, and it was called Vectibix, and it was an EGFR inhibitor, and its purpose was to help with colorectal cancer. So the drug was approved in the USA. The following year, they presented the same data that the FDA saw to EMEA, the European Drugs Regulators. And what happened there was that the data was rejected by EMEA for a lack of efficacy. And the reason for that was that most of the patients that took Vectibix didn't get much benefit, or indeed any benefit. And I think the response rate was about 16 or 17%. And EMEA felt that was too low in terms of in the light of the side effects profile. So, going back to the objections I showed you on the previous slide, is a companion diagnostic economically viable? Well, at this point, the Amgen had literally no sales of Vectibix in the whole of Europe because they didn't have a diagnostic test to improve things. And the reason they thought that they might have a diagnostic test is that prior to this work, or prior to these dates rather, there had been some small studies kind of indicating that mutations in the KRAS gene may predict response to Vectibix. And that makes an awful lot of sense because EGFR is the receptor on the outside of the cancer cell, and it starts a pathway which then transfers a signal down to KRAS, which is part of the pathway. And if the KRAS gene's mutated, KRAS protein gets locked into the on position. So you would imagine it would make sense scientifically that if KRAS was mutated and switched on, there would be no point in switching it off upstream. So that it had quite a good rationale. So Amgen decided they would look into this closer, and it turned out that we were one of the very few, if not the only company, I think very few it was actually, company that had a KRAS test in the marketplace. We had quite a good KRAS, which we'd made as a research use only test. And Amgen approached us to sponsor development of a KRAS test that would be CE marked, that would meet the European diagnostic regulations, and it could be marketed alongside Vectibix. And of course, we were delighted to do this, and we developed the test. And Amgen used that test in a clinical trial of 450 patients. And the results were quite striking, that if you had a KRAS mutation, you didn't get any benefit from Vectibix. It was as clear cut as that. About 40% of colorectal cancer patients carry a KRAS mutation. So we'd managed to take, of the 100% of possible patients, we'd managed to get rid of 40%. So in the 60% that was left, that's where the efficacy remained. And that meant that the efficacy was better and the drug could be approved. And that's exactly what happened. So the EMA approved Vectibix in Europe for patients whose tumour do not carry a KRAS mutation. And then we went further. We managed to get the drug and the diagnostic launched at the same time. And that was a really important thing to do. That, you know, the drug companies rely on the diagnostic companies to do this stuff on time, to get it technically ready, to get manufacturing ready, and to get regulatory approval all done in time to allow a joint launch of the products. And then, as you'd expect, there was further conversations between Amgen and the FDA about how to take things forward in the USA. And then, quite conveniently, as far as we were concerned, there was another drug called Erbitux, which had a very similar mechanism of action, and of course, it relied on KRAS status exactly the same way. So that was also included in the drugs that the KRAS test would test for. And then after that, we actually got acquired by QIAGEN. So if you know QIAGEN, the QIAGEN personalised medicine portfolio was built upon this work and other tests that DXS brought into the fold. So that was a great success. I mean, it worked really well. The two companies worked together, and we managed to do what we set out to do. So I think a lot of the concerns that people had about, is it possible? Is it too difficult? Is it going to cause a problem? Was sort of reassured by that. Now, not surprisingly, so that was one example of a drug and a diagnostic. And what I've tried to do in this slide is just to split down the sorts of types of drugs that you could have that you might want a companion diagnostic for, and also the impact that the companion diagnostic could have. So I've split the drugs into two different types, either branded pharmaceutical products that are coming to market or still on patent, or generic pharmaceutical products that are off patent. Maybe it's not as valuable to the drug companies as the branded ones. And then what could the companion diagnostic do? Well, in the case of Vectibix, the example I've just showed you, it permitted sales. So without a companion diagnostic, there wasn't any sales. It could increase the sales. So it may not be necessary, but it may be helpful. It could be that the companion diagnostic doesn't actually increase the sales, but it identifies a safety risk, and the numbers stay about the same, but the drug gets safe. If it's a rare safety risk, or it may be that actually what the diagnostic does is identify a whole set of people who shouldn't be taking this drug in the first place, And what you're doing is not increasing sales, you're saving money. So there's different types of this. So you've got branded and generic drugs and different effects of the companion diagnostic. And not surprisingly, the place where we've seen most, certainly not all, but most of the successful co-developments over the years is to the areas where it's a branded pharmaceutical product and where you are actually either allowing or improving the sales of the drugs. So we need to think how the diagnostic and the pharmaceutical industries work together. So I've got these two diagrams to illustrate the relative scale and size of the diagnostic and pharmaceutical industries. And I've represented the diagnostic industries here by a big spaceship. It's flying off into space over a planet, and you see it's got a lot of boosters at the back. It looks like a pretty big spaceship, and it is a big spaceship, but it's nothing like as big a spaceship as the pharmaceutical industry, which is following close behind. So to put that into more quantitative terms, the pharma industry is about a $1.4 trillion industry, and the diagnostics industry is about $62 billion. So $62 billion, it's a big industry, but it's about 200 times smaller than the pharmaceutical industry. And the other thing that has a big impact on how this all plays out is the value of the products. Pharma drug products tend to be $1 billion or $2 billion or $5 billion. That's the sort of revenues you might expect to see from a drug in a year. But diagnostic products can be much smaller than that. $5 million to $100 million is a kind of the range for the annual sales of a diagnostic. And the way that the diagnostic companies deal with that is they tend to have a portfolio of many products, each one doing not as many as a drug, but building up to make a reasonable figure. So the pharmaceutical industry, it's a target discovery, then looking for a lead, then looking for some sort of effect. If it looks promising, taking through to clinical development, approval, and launch. It's a slow, risky clinical development process, and it's based on a deep understanding of biological processes to generate a small number of high-margin products. It's really based on biology and science. Diagnostics industry is a little bit different. This is kind of how the things flow in the world of diagnostics. You start with a doctor and a patient. It's often a blood sample. It doesn't have to be a blood sample. Doctor orders a test. The blood sample goes to a lab. The lab do the test. The test result comes back to a doctor. So the place where the diagnostics get done is normally, not always, but normally a laboratory. And supplying those laboratories are diagnostic companies. And they supply two things. They supply the platform, which is the machines and the instruments, which is going to do the testing, and the content. These are the kits or the reagents that are going to go on the platforms. So thinking about what the industry looks like, it's comparatively rapid development. It's lowish risk technical process. Generally, if a diagnostic company starts with a biomarker in mind and has the technology, it's going to be able to make the product. It's based on an understanding of chemistry and engineering to generate a portfolio of mid-margin products. And biomarker discovery is not a major focus of many diagnostic companies. There's much less medicine and clinical chemistry, not clinical chemistry, clinical aspects within the diagnostic industry. In terms of how it breaks down into different types of diagnostics, the biggest sector by quite a long way is the centralized laboratory. It says here CCIA, clinical chemistry and immunoassay. Big instruments doing very routine tests in many, many hospital laboratories around the world. Then there's other sections, microbiology, hemostasis, hematology, tissue testing, which is immunohistochemistry and cancer samples. And then molecular testing. And molecular testing is the second biggest segment of the market. So it's, in terms of how the industry is organized, for most segments, the systems are closed. And what that means is that the in vitro diagnostic company produces the platform, the machine, and the content. and they don't allow third parties to develop onto that platform, which limits the options. So for any drug company looking for a companion diagnostic partner in those segments where it's closed systems, they've really got to go where the systems are. It's the molecular segment that's unusual in that the platforms are often decoupled from the diagnostic content. For example, the manufacturers of DNA sequencing machines are not also the manufacturers of all DNA sequencing diagnostics. You're allowed to develop your own test to run on someone else's machine. So it's maybe fortunate that a lot of the developments in companion diagnostics tended to be in the molecular section, where there was a lot more freedom and a lot more opportunities for innovation, which I think has worked out well for everyone, really. The other thing to be aware of as you think about diagnostics is the difference between diagnostic products and diagnostic services. So most diagnostic tests, as I said, are performed in laboratories, and this is called providing a diagnostic service. The sample comes in and the service is to do the testing. So thinking about how a lab is going to introduce a new test, that they have a choice. One option, and the most common one, I think, is to purchase it already developed by a diagnostics product company. So you buy the test, you buy a kit, you put it in, it's already been approved, it's already regulated, it's in the lab and it's up and running. The alternative is to develop it themselves as a laboratory developed test. A laboratory developed test is exactly what it says. The laboratory goes about and identifies the reagents, it buys in some equipment and takes responsibility for developing its own test. So from a companion diagnostic perspective, both of these are possible. A drug company could contract with a diagnostic company to build a product to sell to many labs, or it could contract with a single lab to develop a laboratory to develop a test. Now, so from a companion diagnostic perspective, LDTs, as they're called, laboratory developed tests, they can be quicker and cheaper to develop than an in vitro diagnostic product. That's partly because within a laboratory, you've taken a lot of the variation out. As you develop in a product, you have to develop a product that's robust enough to work not in your lab, but in anybody else's lab who happens to buy it. With a laboratory developed test, you're in a much more controlled environment. But the downside is, and that's the biggest downside, is LDTs are geographically limited to the lab, to one lab. That means that the samples have got to travel. So if you can't move the test to the samples have got to move to the test, which is bad enough, shipping them across the whole of the USA. But when you've got to start doing international shipments at all, it can really slow down the uptake of the test, which of course is the last thing that the drug companies want, who want these things to be readily available. Also, the LDTs are not typically regulated to the same standards as IVDs. And whereas it would work fine, I think, in terms of bringing a new test to market, if I'm a drug company, I don't want any regulatory ambiguity. So for those reasons, most drug diagnostic companion diagnostic partnerships are to develop and gain regulatory approval for companion diagnostic products. The other big factor that has an impact on how all this works out is the companion diagnostic clinical trial. And a companion diagnostic is expensive because it's essentially a drug trial. You're not trying to show that your test works. You're not saying I can pick up this biomarker. You're trying to say I can pick up this biomarker and it's useful in terms of patient outcomes. And the appropriate samples to test the utility of a companion diagnostic. And from a group of patients all taking a therapy, well, only some of them responding well to it. And the people that have those samples are, of course, the drug companies. So from a diagnostic company perspective, it's difficult to justify the cost of setting up a companion diagnostic clinical trial, just in terms of the revenues you're going to get back from the diagnostic product. So these things tend to be set up by the drug company. And of course, the pharmaceutical company, diagnostic company partnership solves this problem that the drug company set up the trial, the diagnostic companies provide the test. And there's typically a three-stage roadmap in the life of any companion diagnostic. And you can see this from the example I gave earlier with KRAS and Vectibix. But you could also, if you're familiar with other examples, I'm sure you could apply this roadmap. The first thing is to identify the biomarkers for the test and then get the drug into development. Actually, identifying the biomarkers is not necessarily or even typically done by the diagnostic company. It could be done by the drug company, or it could be subcontracted by the drug company to someone else. Having some biomarkers is the first thing to do. You need the biomarkers are then translated into a test by the companion diagnostic test by the diagnostic company, whereas the drug goes into development in phase 1. What you really want to do is speed up that diagnostic development so that the companion diagnostic test is ready for phase 2, possibly in phase 3, certainly, so that you're using the same test during the clinical trial as you're using during subsequently when the drug gets onto the market. And then once that all works out well, the diagnostic company sells the diagnostic and the drug company sells the drug. And that's the model, and that works well. That's worked well over the years. So in terms of success factors, what's important for this to work? The drug company partner is in the best place to identify the biomarkers as they have access to the appropriate samples. With a new drug, no one else is going to have access to patients responding well or badly to it because by definition it's a new drug. The drug company partner takes responsibility for the clinical trials and they sponsor the development of the diagnostic test. And this is, of course, why diagnostic companies really like this model, because when we say sponsor, they mean pay for it. So money changes hands from the drug company to the diagnostic company to make this happen in a timely fashion. From the diagnostic company's perspective, they have to make sure the test is available for use in the clinical trial. Its performance has been shown by analytical validation. There is regulatory approval, and the test is available and can be distributed. And that diagnostic drug company partnership is now firmly established as a business model. I went on Genome Web and I just put in the XRX partnerships in the last couple of months, and there's loads of them. I mean, it's a very popular, it's a very successful model. And in terms of those barriers that I talked about back from the early 2000s or the mid 2000s, is it needed? Well, of course it's needed, because you can't sell your drug without it nowadays. It's not economically viable. I think there's been a big switch in the pharmaceutical industry. I think they're happier to have much more efficacious drugs working in smaller populations and possibly getting paid more for them, and that's really worked out. So that's good. I mean, that's good for drug companies, but it's also very good for the patients. Is it too difficult? Well, no, it's not too difficult. There's loads of examples of it now. And of course, now there's hundreds of precedents for this. So I think a lot of the concerns have gone. So we've talked about branded pharmaceutical products. We've talked about where branded pharmaceutical products need a diagnostic to allow it to get to market, and some of the commercial forces, and how the importance of getting a successful drug drives forward the development of the companion diagnostic. What about the other situation? What about when you've got a generic pharmaceutical product? Maybe we're not going to increase the sales. Maybe we're going to improve the safety or something like that. How does that work? Do we have some examples of that? Well, yes, we do. And I'm going to give you an example from Yourgene Health, which is the company I founded after DXS. I'm still there in an advisory capacity. And a test that we developed called for DPYD deficiency. And where this is important is if you're taking the drug 5-fluorouracil, which appears in several different guises, and it's a chemotherapeutic agent for use in treatment of a range of cancers: colorectal, gastric, breast. And it's estimated that about 2 million people are treated with 5-FU every year. Now, of these, 10 to 20% will be hospitalized because of 5-FU toxicity. It's not a safe drug. Like a lot of cancer drugs, it's not a safe drug. And 0.1% maybe these people might die. We know the reason for part of this toxicity. We all carry the DPYD gene. Some people carry a single mutation in it. Some people carry 2 mutations in it. And I'll show you why that's important. There's an enzyme, thymidylate synthase. It converts deoxyuracil monophosphate thymidine monophosphate. 5-FU inhibits thymidylate synthase. 5-FU in itself is broken down by we'll just call it DPYD, just not be having to say a lot of complicated words. Makes the DPD product. If you have a mutation in your DPYD gene, you don't make the DPD product, you don't break down the 5-FU, and you essentially get an overdose of 5-FU, which can be toxic and even lethal. This has been known for a while. Here's an example of a trial that took place in 2017. I think it was a good trial. Prevention of 5-FU induced severe toxicity. Clinical utility within this trial with DPDY genotyping. They got 11% early tox and no deaths out of 1,000. Without it, they got 17% early tox and 23 deaths. And they had to stop the trial on ethical grounds. You can see it's very effective. It's a very effective test in terms of increasing safety profiles. The challenge is that no one really took on this clinical trial work until it was done by essentially a public funded body. And what's been very helpful in terms of bringing DPYD tests to market is that some other organization has taken on the role of the pharmaceutical company. So not, in the first examples I showed you, the drug companies took on the responsibility for the clinical trial. Here's the clinical trial has been publicly funded. And the other thing that was very helpful is the Clinical Pharmacogenetics Consortium have pulled all the information from this trial and all the other ones together, and they've come out with a set of guidelines called the CPIC guidelines, which tell you what to do when you find a 5-FU mutation. The other thing that's kind of driven this is some adverse publicity. This is a quote from the Daily Mail, not the UK's most salubrious newspaper, but a perfectly respectable one, revealed how hundreds are being killed by a chemo meant to save them, and it's down to a little-known side effect which can be avoided with a simple £60 blood test. And USA Today have got a similar story there, and here's a sad story from Oregon Health and Science University, who was sued because a patient died and the test wasn't available. So all these things come together: the science, the clinical trial work that various people have published, the CPIC guidelines, and the publicity led Yourgene Health to put together its own Yourgene DPYD in vitro diagnostic. It's a regulated, it's approved, it's available in Europe, and that's been very helpful. So, non-commercial factors in the introduction of personalised medicine. So the things that made a difference: publicly funded studies, expert panels, public awareness. So eventually, even though no one made any money out of this companion diagnostic, except for Yourgene a bit, I hope, certainly not the sort of revenues you would get associated with a therapy, it still was successful and it's still got there. Well, I want to show you this particular paper: life-threatening toxicity in a dihydropyrimidine dehydrogenase deficient patient after treatment with topical 5-FU. So this was the first report of exactly what we're talking about, the DPD deficiency and the bad side effect. And look at this. It was August 1999. So I think it shows you that we're quite capable of bringing companion diagnostics to market for generics as well as ethicals, But gosh, it's a slow process. And I think, whereas I think we can be proud as a scientific and clinical community about the way that companion diagnostics and personalised medicine has really taken off in some fields. There's still a lot of work, I think, to be done in these in drugs like 5-FU, and there's certainly some more examples of them. A little detour here. We do DPYD testing. If you'd had your whole genome sequence, you would have had that information. And for at least the last 30 years, hundreds have confidently been predicting that individual genetic tests would become redundant. And at first, the expectation was that we'd all be genotyped, and that lasted from about 1990 to 2010, or we'd all be sequenced at birth. And generally, when people suggested this, we said that we would carry the data around with us on a credit card-sized device. And it certainly hasn't happened. I was thinking about why hasn't it happened? I don't know, maybe therapies and interventions need to be better. I was thinking, imagine you got yourself genotyped, someone's telling you you're at risk of Alzheimer's, you've got an ApoE, you're an ApoE homozygote. That's a tricky piece of information to deal with in the absence of anything to do with it. Once there's a treatment available, doesn't that change things? So maybe some of this whole genome sequence to identify unknown traits and predispositions will be driven by therapies. The other thing is probably better polygenic risk scores. At the moment, we can do a risk score, but they're not as good as they could be. I'm sure that will come with time. So maybe this will change over time, but at the moment, I don't see the individual tests for DPYD or KRAS or EGFR or anything else like that disappearing in the short term. I've got a few minutes left. I just wanted to finish off with a few technology trends that are likely to impact personalised medicine. They're actually likely to impact molecular diagnostics and diagnostics in general, but they'll certainly have an impact on personalised medicine. One of the things I think is an increased range of samples. We're very good at testing blood. Blood's a great sample, and if you want to look what's going on in the body, blood's excellent. And if you want to know what someone's genome is, using a cheek scrape or spit or saliva is a very convenient sample. I mean, there's other samples, there's urine, the stool. I think the sample type that hasn't really taken off yet, but I predict that it will, is VOCs, volatile organic compounds in breath. They sample what's in the lung. There's a vast variety, very rich data source, but they haven't really taken off yet. I think one of the reasons for this is that it's actually surprisingly difficult to get a clean sample. There's an awful lot of risks of contamination. I'm working with a company, Bioxhale, and they're really focusing on how do you take a sample? How do you take a sample? How do you make it easy for the patient? How do you make it easy for the nurse? And how do you keep it clean and not contaminated with the environment? So I'm expecting breath sampling to become an important trend in the next year or two. I think we're going to see combining different types of samples in analysis. Cell-free DNA is a fantastic biomarker. It's very important. It's very simple and convenient. But often, just looking at cell-free DNA lacks the sensitivity and specificity needed to be a reliable, particularly early detection marker for cancer. But other assays are available. We can look at cell-free DNA, we can look at specific biomarkers for cancer. We could look at things like proteomics or metabolomics. I'm working with a company called Dxcover, who uses Fourier transformed infrared spectroscopy to generate a profile from blood which shows different patterns in cancer patients and non-healthy populations. So I would expect that we might expect to see these different modalities added together to run 2 or 3 different types of tests, which in totality may allow us to have more powerful screening and diagnostic tools. Home testing, I think, could become important. At present, the sort of diagnostic tests that can be performed at home are rather limited. But I think technologies emerging which will allow regular quantitative monitoring of selected biomarkers at home. And I think that could be so important because I think a one-off test will tell you a point in time Testing it every day or every week or even every month will give you a pattern of normality, so you can see what's abnormal. So I think that's going to be an important trend over the next few years for diagnostics. For companion diagnostics, I think it's important because the drug companies don't really like being in thrall to the testing lab and all of having to rely on all of that diagnostic infrastructure. Taking back control through having a home testing device could be quite attractive from the pharmaceutical company. So I wouldn't be surprised if we see some companion diagnostic partnerships based around home testing technology. And the other thing I'm expecting to see is that technology may move from analog to digital. What I mean by that is that the moment diagnostic testing technologies are based on the measurement of populations of molecules, because a single molecule can't generate enough signal to be visible. And some methods use clonal amplification to take a single molecule, make a clone of it, and build thousands or millions of copies of it and test that. So next generation sequencing or droplet digital PCR would do that. They have their value. But now a company like SMI, who I know, and I'm sure other companies as well, are developing methods which are so sensitive they can dispense with the amplification step and just get down to single molecules and look at those single molecules directly. Once we can do that, it's going to allow much more accurate quantification, and it's also going to allow lower limits of detection, which could become very important in terms of early detection, screening, and monitoring of cancers and other diseases. So I'm going to come to a close there. The last 20 years has seen very many examples of pharma company, diagnostic company partnerships which have improved healthcare by bringing drugs and their companion diagnostics to market. So I think that's a good news message. The model pioneered by DXS in 2007 is now firmly established worldwide. So I'm pleased that we were helpful in regard with that. Despite these successes, there's been rather less progress with generic drugs, safety biomarkers, and companion diagnostic tests, which can save money rather than improve outcomes. I think there's room for improvement here, but I'm sure that will take place. Recent developments, and it's shown, of course, by recent developments like DPYD, which shows that introduction of these tests is feasible. It just takes a lot longer. And then the field will likely be transformed again. I don't know how much it's going to be transformed to some extent as new technologies come online. Some of the technologies I mentioned today, but I'm sure there's many technologies which I don't know about. Thank you very much. Goodbye.
Thank you, Dr. Stephen Little, for your informative presentation. We will now start the live Q&A portion of the webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. All right, so let's get started. Our first question here asks, Dr. Little, you mentioned a few technology trends that might be important in the future. Do you think cell-free DNA will be an important methodology for personalized medicine?
Yes, in a nutshell, yes, I do. Though there's an important limitation with cell-free DNA, and the problem is one of lack of molecules. So you might, in 10 mils of blood, you might get around about 20 nanograms of DNA. A diploid genome weighs 6.5 picograms. So in 10 mils of blood, you might get 1,500 genomes worth of DNA. Now, you've got to think about what does that mean? 15 with that number of genomes, 1,500 genomes. That's plenty to work with. You could measure something in 1,500. But often when we talk about cell-free DNA, we're interested in looking at cancer. So the bit of the cell-free DNA that's relevant is the cancer fraction. And given that the tumor may be small or it may be very small, particularly if we're using it in an early detection, or a pre-symptomatic way, the cancer fraction could be 1% or less. And with 1,500 molecules and a 1% cancer fraction, it means in 10 mils of plasma, 10 mils of blood, you've got about 15 molecules. And the lower cancer fractions, the number of molecules might be less than 1. Now, the problem with having molecules less than 1 is that it doesn't matter how clever your analysis technology is, you'll never be able to see them because they aren't there. So one of the big limitations of cell-free DNA at the moment is number of molecules. So I think we're interested in seeing what technologies or techniques could be used to increase the number of molecules. Well, I've seen some different ideas. One idea is that people could take a drug or a medicine that would sort of loosen up the amount of cell-free DNA and increase it in the blood. I think that might work technically, but I think it sounds like a dangerous sort of drug to take. I don't know. It may turn out that it's perfectly safe, and that's the way things end up. But the idea of having an injection or something that then increases the amount of cell-free DNA in your blood doesn't sound to me to be a particularly promising idea. So the idea that I've been following is a different one, and it's based on sampling a lot more blood. So we could take 10 mils of blood, we could probably take 20 mils of blood, but you can't take pints of the stuff. It isn't going to be practical. So I've been working with a company, and what they're doing is they're taking the blood out, they're running it through an apheresis machine, they're taking the plasma that's created and running it over a DNA collection matrix, which takes the cell-free DNA out, and then it goes back into the body. And I think this might become a very important technology, and that it could, cell-free DNA in personalized medicine and companion diagnostics has already got a lot of utility. There's no question about it, but it's limited by these sample amounts. I think if we can crack that sample amount problem, it could make a big difference to the uptake of this technology. So I think that may become something that's very important in the next year or so. So time will tell. We'll see.
Absolutely. Well, thank you, Stephen. We are running low on time here, so I think that we are going to go ahead and wrap up after that question, but we do appreciate you being here for this live presentation. Stephen, do you have any final comments that you'd like to provide our audience?
Not really. Maybe just that, I kind of said it already in the conclusions, but I guess I'm a naturally optimistic person, and it's easy to hear bad news all the time, but I think what we've done with personalized medicine is a great success so far. Well done to the clinical, the scientific, and the technical communities for making this happen. And I'm sure we're going to see more examples of this moving forward, which will benefit people commercially. It'll be useful for business, but primarily it's better for the patients, and that's a great place to end this talk, I think. Thank you very much.
Wonderful. Well, thank you again. I'll provide one more final thank you for you, Dr. Stephen Little here. Thank you for your time and your important research. This does conclude our session today, but before we go, I would like to thank the audience for joining us and for their interesting questions. Questions we did not have time for today, and those submitted during the on-demand period will be addressed by our speaker via the contact information you provided at the time of registration. This webcast can be viewed on demand for 2 years until May 18th, 2025. Labyrinths will alert you via email when it's available for replay, and we encourage you to share that email with your colleagues who may have missed today's live event. So until next time, take care, everyone. Goodbye.